Hi, everyone, and thank you for being here today, and certainly to the ARM organization for hosting us. My name is Lisa Deschamps. I'm the CEO of Aviado Bio. We are a clinical stage CNS gene therapy company. We're London-based, but of course globally focused. A little bit about Aviado Bio. Our mission is to bring curative gene therapies to those living with neurological conditions. We are starting this effort in neurodegeneration. I'll tell you much more about that in a few minutes. And overall, our approach is all about drug and delivery. So taking therapeutics that we know a lot about, like AAV9 and microRNA, and coupling them with novel direct delivery approaches, really very much focused on how do we address some of the key challenges that exist today across CNS diseases. We have a number of different programs. Our lead program has just entered the clinic, and we're now dosing patients. This is a program in FTD, frontotemporal dementia, in patients who have a monogenic form of the disease, and we'll share more about that. And our pipeline is very much focused on ALS. From a team perspective, we have about 60 people today in London, and again, we're very much focused on executing our programs all around the world. Our funding to date has been, collectively, we've raised $96 million. Our seed round back at the end of 2020 was $16 million. In 2021, we did an A round of $80 million. And we've been very much focused on bringing these programs to the clinic as quickly as possible for patients in need. We have a very experienced team. I think all of us are only as good as our people. And I'm very proud to say that everybody from the labs all the way through to the C-suite has gene therapy experience. Everyone has CNS gene therapy experience, and many of our team members have worked across multiple therapeutic areas and organs in the space and have been in the cell and gene therapy space for a number of decades. And I do think that's what's allowed us to move so quickly from proof of concept only at the end of 2020 to now clinical stage, in early 24. The company is a spin-out of Professor Chris Shaw's lab at King's College London, so the science was originated in the UK, and Chris is a neurologist, neuroscientist who's been treating forms of dementia and ALS for over 30 years, so we're very much founded by one of the world's leading experts in these disease areas. We have a syndicate of 8 top-tier blue-chip investors who are very much also heavily invested in cell and gene therapies and have very much supported us from the original seed round all the way through and continue to do so. A little bit about our pipeline. As mentioned, we are very much focused on drug and delivery. So all of our programs not only have the therapeutic focus, but a novel direct delivery approach. Our lead program, which has just entered the clinic, is AVB-101. Again, this is a program in FTD for patients who have a monogenic form. It is a gene supplement program, and these patients have a specific mutation in their GRN gene. So we're looking to restore and supplement the progranulin levels to as close to normal as possible. This program is also, from a delivery perspective, intraparenchymal, so novel direct delivery into the brain and specifically into the thalamus, and I'll tell you why that's important in a second. On the pipeline side, we are focused on 2 targets in ALS, ataxin-2, which basically represents 97% of all of ALS, as well as is responsible for a number of other neurodegenerative disease, and SOD1, so both sporadic and non-sporadic disease. These 2 programs have a novel direct delivery approach called SubPeel, which is direct into the spinal cord, and it's a platform that we have exclusive rights across CNS therapy. I'll show you what that looks like. We also have a number of other undisclosed targets that we're looking at across neurodegeneration. A little bit about FTD, if you don't know. Unfortunately, recently this has gotten some publicity with some very famous celebrities being recently diagnosed. One is Bruce Willis, who has a language form, PPA, and unfortunately just recently Wendy Williams. Despite just really coming to the forefront recently, this is one of the most common forms of dementia in people under the age of 60, so very much affects younger people in the prime of their lives, very much contributing to society. We have met a number of families, unfortunately, as early as 25 and 30, already symptomatic, not just gene carriers. FTD, from prevalence perspective, just in the US and top 5 EU countries alone has about 100,000 patients today that have been diagnosed. In this particular monogenic form we're looking at, it affects about 10% of FTD, and there's about 1,600 new patients every year diagnosed with this disease. It really affects your whole being, so it's not just how you would think about Alzheimer's, where it starts out cognitively and then obviously affects all bodily function. This disease really changes the person you are. So very much in many cases affects personality, behavior, communication, speech, and then takes on your full kind of executive function, and of course also includes then the more common forms of dementia like cognitive impairment, et cetera. It is a very fast-moving form of disease, which is terrible, of course, for the patients and families affected. From a clinical trial perspective, it's very tractable and easy to study. From a disease biology perspective, we do understand it, especially the monogenic forms. We understand the root causes. From a progranulin perspective, for you and I who don't have this form of disease, we have about 50% more than these impacted patients of progranulin levels. When progranulin decreases, TDP-43 accumulates, and that's what causes the neurodegeneration. So our goal is to restore as much progranulin in these patients to as close to normal as possible. So not only we can prevent, but potentially even reverse for some of these patients whose neurons haven't yet died. It is very tractable from a clinical development perspective, so there is natural history cohorts, there are established registries both in the US and Europe. So again, from a trial perspective, while of course we always want more, it does give us a very good starting point and foundation to understand the disease and be able to really study and follow it. A little bit about why we chose our delivery approach. So again, intraparenchymal direct into the brain, but specifically the thalamus. Thalamus is deep in the brain structure and pretty much a sensory relay hub to the brain. So we're really using the thalamus to do the work for us. And by targeting the thalamus, we can use very low doses of treatment in a very targeted way, and then allow it to kind of distribute for us to the rest of the cortical regions that are needed to potentially, again, treat this disease. We do have a very extensive and robust preclinical package across, we've looked at over almost 60 animals across mice, sheep, and NHPs from a regulatory enabling study. It is one of the most robust packages in this space. Obviously, we've been able to generate all of this data over the last couple of years. A little bit about our clinical trial. It is an open label dose escalating phase 1/2 study. Primary endpoint is safety. We're looking for 6 to 9 participants for this phase of development. The difference between 6 or 9 will just depend on the dose, right, and how many patients we can dose at each of the cohorts. We'll be treating patients. We've now started 9 weeks apart, and hopefully this year, assuming all goes well, we'll look to treat 3 or possibly even 4 patients. And then from an endpoint perspective, it is a phase 1/2 study, so of course primary endpoint is safety, but these are the different markers that we'll be looking at across the development phase of this study. So really once you get started, data will start to roll in. It's open label. We'll be able to really focus and monitor these patients in real time as we go. Acute safety, so this is a neurosurgical procedure, so we'll be able to assess acute safety within the first 2 weeks of dose as it relates to the surgical procedure. Then, of course, from a biomarker and clinical perspective, we'll be looking at progranulin levels in the CSF. We've done baseline and then every month, and then we'll batch them at 3 months and start reporting out the data. From a primary endpoint, that is the 6-month safety endpoint. And then, of course, we will start to follow patients from looking for functional and clinical efficacy benefits over the course from 6 months onward. So looking at volumetric MRI, tracking atrophy, and other biomarkers like neurofilaments. And then there's a number of clinical scales that will really look for functional and efficacy benefits in these patients that will be tracking 12 to 24 months. And the study follow-up will be 4.5 years. So we will have quite a robust data set in this study. It is a stereotactic procedure, so we'll be under MRI guidance. We're using the ClearPoint system, which is an established system. Many of you might be familiar with it. In the US alone, there are almost 70 centers that have ClearPoint already installed. In Europe, all the key markets have at least 1 to 2 centers, and that is really expanding in real time. And this allows us to really be able to, see where the vector is going and make sure that we're getting the coverage that we need to ensure that we can address the disease as we go. This has also been done. Intraparenchymal delivery is becoming much more mainstream. So PTC has recently got approval in Europe and AADC. It's now pending in the US. And so I think we will see a lot more of this as we go. There's a number of other industry partners, some that are here, that are also looking at intraparenchymal delivery. Because of the nature of the delivery approach, it is a neurosurgical procedure. So as you would imagine, it's a hub and spoke model where we have, there's a referring neurologist, a treating neurologist, and then a neurosurgeon who will actually conduct the procedure itself. And obviously, it's a very tight network that will be working across to ensure that we take good care of these patients along their journey. We do have 8 countries that now are approved for this global study. So we submitted to 9 regulators and got 8 approvals. So I think that's a pretty good success rate. This includes key markets in Europe, UK, as well as the US IND is now open with fast track designation. We are recruiting currently in 3 of these countries, Poland, Spain, and the Netherlands. The US will be open in the next month, so mid-May. And the other 4 countries will be open in a staggering effect. They were just going through logistical IRB reviews, site contracting, et cetera. A little bit about the portfolio. As mentioned, we're very much focused on ALS. We have also a novel direct delivery approach, which goes direct into the spinal cord from a subpeel perspective. I'll show you what that looks like in a second. We are this year going to get into we've just done candidate nomination, and we're going to get into our GLP tox, our regulatory enabling studies, and hope to get into the clinic as early as end of 25. Those of you probably much more familiar with ALS, so I won't cover it as we don't have too much time to do that, but of course also extremely debilitating from the onset of disease to death can be as early as 2 to 4 years, so a very fast-moving, unfortunate disease. While there's been some progress in this field, there's still tremendous opportunity to bring a cure and something that will truly transform the care for these patients. We understand the disease biology there. There's some established endpoints and clinical markers that we can really look at and be able to track. And there's there's a tremendous amount of advocacy in this space and, natural history and a lot that we know about this disease. We just need the treatments to bring to truly, transform the care in this space. I'm really excited about the work we're doing. I think we have a very good shot at doing it. We're using AAV9 in both of our programs. In our ALS pipeline, we're also coupling that with a proprietary microRNA platform, which is very targeted and efficient and becomes really a delivery vehicle in of itself. A little bit about our delivery approach. So this is a subpeel delivery. We have licensed this technology from a group out in California. From USD, and it's really meant to again address the key challenges that exist in neuroscience today. Your peel membrane covers your brain and your spinal cord, kind of as a saran wrap around, and it's really hard to penetrate and get anything in, which is why you see very little transduction when you use intrathecal, why many of our peers, including products I've formerly worked on, you have to use very high doses to get anywhere into near the brain or the spinal cord, and then you result in, unfortunately, often toxicities that don't, outweigh the clinical, that overcome the clinical benefit. Subpeel has been studied. While it's new and hasn't been used in the clinic from a preclinical perspective, there are a number of papers that are out there that the inventor, Martin Masala, who he's licensed this technology from, has put out there. And you can see here, just again, looking at, this is not our construct, but looking at subpeel delivery versus intrathecal, you see a huge difference in transduction compared to intrathecal. And again, we're hoping to do very similar to what we're doing in our lead program, be able to use very small doses in a very targeted and efficient way that will really translate to maximal therapeutic benefit for patients in the safest way possible. This is just a picture of the platform. There's a proprietary needle, and it's really very similar to laminectomy, so it's very quick and easy to train on, and so it's very scalable in the real world. So in summary, I want to thank you for your time. Again, we are very much focused on drug and delivery. We have a program now in the clinic, and we are hoping to get our pipeline assets as close to the clinic as possible by the end of this year. Thank you for your time.